Welcome to Roadmap to Residency, Evidence-Based Applications. Hello, everyone. Welcome to our Club 250 Matchmaker sessions. So in this video, we will be discussing about glycolysis. So glycolysis, so it is used by all cells of the body. Sequence of reactions that occurs in cytoplasm converts glucose 6 carbons to pyruvate 3 carbons and generates ATP and NADH. So NADH, which is also called as a nicotinamide adenine dinucleotide, have two nucleotides, and it carries electrons. Carries means either it can accept electron or it can donate the electron. The acceptance of electron is by NAD. When it accepts electron, it is called as NAD plus, and when it donates electron, it is called as NADH. So this is the basic pathway of glycolysis. So glucose gets converted to glucose 6-phosphate, which gets converted to fructose 6-phosphate, which gets converted to fructose 1,6-bisphosphate, then glyceraldehyde 3-phosphate, which combines with dihydroxyacetone phosphate. Okay, so it can form glyceraldehyde 3-phosphate plus dihydroxyacetone phosphate. This dihydroxyacetone phosphate is not generally an important part for the examination, and again that forms 1,3-bisphosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, phosphoenol pyruvate, and at last it forms pyruvate. So hexokinase versus glucokinase. So hexokinase is found in most tissues, is strongly inhibited by glucose 6-phosphate and blocks the cells from holding glucose. Insulin has no effect on hexokinase, and it have low Km, which usually operates at max, and it has low Vm, so maximum is not that high. So it has low Km and low Vm. So glucokinase is found in liver and pancreas, not inhibited by glucose 6-phosphate, induced by insulin. Insulin promotes the transcription and inhibited by fructose 6-phosphate, and it has a high Km. So the rate varies with glucose. So glucokinase is regulatory protein, or GKRP, translocates the glucokinase to nucleus. Result is inactivation of enzyme. So fructose 6-phosphate. So GKRP binds with glucokinase, and then the nucleus becomes inactive. In case of glucose, it competes with GKRP for GK binding, and in the glucokinase, it goes to cytosol and becomes active. So here we have the glucokinase, which binds with GKRP, okay? And again, by the enzyme fructose 6-phosphate, okay, it goes into the nucleus where it becomes inactive. So in case of low blood sugar, the hexokinase working. The hexokinase is working and glucokinase is inactive, okay? And glucose to the tissues, glucose goes to the tissues and not to the liver. In case of high blood sugar, the hexokinase is inactive and glucokinase is working, and liver will store glucose as glycogen. Now this slide is very, very important. In case of glucokinase deficiency, which results in hyperglycemia, pancreas is sensitive to glucose, and there is mild hyperglycemia, which is often exacerbated during pregnancy. Phosphofructokinase 1 is the rate-limiting step for glycolysis, consumes second ATP in the priming stage, and it is an irreversible step. It commits the glucose to glycolysis via HMP shunt. Glycogen synthesis is no longer possible. Regulation of glycolysis. The key inhibitors are citrate and ATP, and the key inducers are AMP and fructose 2,6-bisphosphate. So this is apart from it first stage, so regulation by fructose 2,6-bisphosphate. So fructose bisphosphate is 2 and phosphofructokinase 2 are the same bifunctional enzyme whose function is reversed by phosphorylation by protein kinase A. So here we can see the fructose 2,6-bisphosphate, okay, it can stimulate the phosphofructokinase 1. And when the phosphofructokinase 1 gets stimulated, the fructose 6-phosphate can change into fructose 1,6-bisphosphate. Also, fructose 1,6-bisphosphate can be changed into fructose 6-phosphate by fructose bisphosphatase 1. Whereas talking about the fructose bisphosphatase 2, which can change the fructose 2,6-bisphosphate to fructose 6-phosphate. So you can remember 2,6-B fructose bisphosphatase 2 acts on fructose 2. 2 is gone, only 6 is left. So fructose 6-phosphate, which is active in the fasting state. Whereas in the fed state, okay, where we have actually had our meal, then the fructose 6-phosphate acts upon the phosphofructokinase 2 acts on the fructose 6-phosphate to form fructose 2,6-bisphosphate. Okay, and whenever there is high quantity of fructose 2,6-bisphosphate, you know what happens? It stimulates the PFK1. So in the fasting state, the increase in the glucagon, increase in the CAMP, increase in the protein kinase A, increase in the fructose bisphosphatase 2, and decrease in the PFK2. So there is less glycolysis and more gluconeogenesis. Whereas in the fed stage, there is more insulin, there is decrease in the CMB, there is decrease in the protein kinase A, there is decrease in the fructose bisphosphatase 2, and there is increase in the PFK2. So there is more glycolysis and less gluconeogenesis. So glycolysis, it has the first stage is called the splitting stage. So fructose 1,6 phosphate to two molecules, okay, so of glyceraldehyde 3-phosphate and a dihydroxyacetone phosphate, and this is a reversible for gluconeogenesis. So and then comes the energy state. So glyceraldehyde 3-phosphate, okay, then it forms the 1,3 bisphosphoglycerate, forms 3-phosphoglycerate 2 and phosphoenol pyruvate. It starts with glyceraldehyde 3-phosphate, 2 ATP per glyceraldehyde 3-phosphate. So total per glucose we have from 4 ATP, right? So pyruvate kinase is not reversible and it is inhibited by ATP and alanine and activated by fructose 1,6 bisphosphate. Glucagon and epinephrine, they play a role in phosphorylation, inactivation of pyruvate kinase, and this slows the glycolysis and favors gluconeogenesis. Talking about the alanine cycle, the skeletal muscles can degrade protein for energy. They produce alanine, which goes to blood and liver converts the alanine to glucose. So in the liver, the alanine forms pyruvate, which can go into form glucose. This glucose can go into the muscle and form again from pyruvate and alanine. And again, by the enzyme alanine transaminases, it can transfer to liver. This is called as alanine cycle. So talking about NADH, there is limited supply of NAD plus, so it must regenerate. Whenever there is oxygen is present, then the NADH gets converted to NAD in the mitochondria, and if it is absent, oxygen is absent, then NADH gets converted to NAD plus via lactate dehydrogenase. Lactic acidosis, so if there is decreased oxygen, decreased pyruvate entry into the TCA cycle, there is increased lactic acid production, which will cause acidosis, elevated anion gap acidosis, and which can go to cause sepsis, bowel ischemia, and seizures. So talking about 2,3-bisphosphoglycerate, which is created from diverted 1,3-BPZ, is used by RBCs, okay, because they have no mitochondria and they sacrifices ATP from glycolysis. So 2,3-BPZ alters the hemoglobin binding. So energy yield from glucose. So the ATP generated depends on cells per oxygen. Highest yield with oxygen and mitochondria allows pyruvate to enter the TCA cycle, and it converts the pyruvate or NADH to ATP. So in oxygen and mitochondria, so glucose plus 6 oxygen gives 32 or 30 ATP plus 6 carbon dioxide plus 6 H2O. 32 ATP is in the malate aspartate shuttle, which happens in the liver and heart, and 30 ATP is in the glycerol 3-phosphate shuttle, which goes towards the muscle. So we have talked about the shuttle in the first video. So no oxygen or no mitochondria, then the glucose only forms 2 ATP and 2 lactate. So this is the summary of everything. So 25-year-old woman presents with fatigue, pallor, and shortness of breath upon exertion. She reports a history of intermittent jaundice since childhood, especially during times of stress or illness. Lab tests reveal elevated levels of unconjugated bilirubin and lactate dehydrogenase in her blood. Further investigation reveals a deficiency of an enzyme involved in the glycolytic pathway. Which of the following enzyme is most likely deficient in this patient? Hexokinase, phosphofructokinase, pyruvate kinase, glyceraldehyde 3-phosphate dehydrogenase, and phosphoglycerate kinase. So the patient's symptom of fatigue, pallor, shortness of breath upon exertion, and intermittent jaundice since childhood, especially during times of stress or illness, suggest a hemolytic anemia. Life findings of elevated unconjugated bilirubin and LDH further support this suspicion. So pyruvate kinase deficiency is the most likely enzyme deficiency in this patient, and which is a key enzyme in the glycolytic pathway responsible for catalyzing the conversion of phosphoenol pyruvate to pyruvate. Now, deficiency of pyruvate kinase can lead to decreased ATP production in red blood cells, impairing their ability to maintain their normal shape and structure, resulting in hemolysis. Thank you for watching. Keep studying hard.